Hello everyone. So welcome to another video of inheritance and variation. So as we know that we were discussing in the previous video about the genetic terminologies, who was Gregor Mendel and what was his work related to genetics. And the terminologies were covered in the previous video. If you have not checked out that video, please go through that. Then in this video, we are going to cover why Mendel chose the pea plant, which is known as the Pisum sativum, as his sample for carrying out genetic experiment. Briefly, what did he do in that hybridization experiment? And we will do the first experiment, that is the first cross that he carried out, that is monohybrid cross. Okay. So let's begin. So Pisum sativum is basically the pea plant, which we called in Hindi as the Matar plant or the Vatana Matar plant. You can see in the figure. Now there were certain characteristics of this pea plant that Mendel chose. Out of so many plants available in nature, he chose this plant. Why? So those characteristics you should be aware of. The very first characteristic is that many pea varieties were available. So pea plant has many varieties of it. Okay. For example, green color pod pea plants, yellow color pod pea plants, then wrinkle seed and the round seeds. Flowers of pea plants are different. Some flowers have white color, some flowers are purple in color. So because of these varieties, so it becomes easier to study genetic traits that gets transferred from parents to the offspring. So this is one of the quality or characteristics that this Pisum sativum possess. Also, these are very small plants and they can easily grow. Okay, so this is one characteristics. Another important characteristic that Mendel shows, that they were smaller and they can easily grow. Okay, then they are self-fertilized and they can also be cross-fertilized easily. Okay, so no tension or no problem in fertilization. They can self-fertilize also, and if you want, you can do emasculation and carry out cross-fertilization also. Okay, they have a very short lifespan and many generations you can study in a small time, so you don't have to wait for a longer period of time. You don't have to wait for months or years. Certain plants take years, one full year for their life cycle to complete, but pea plant, their generation is covered in few months, relatively few days, shorter in lifespan. So that is why he chose this Pisum sativum. Okay. So we will just see the 7 contrasting traits. So just to define the varieties or the selected characters that Mendel focused on, and why he chose this pea plant was because of these 7 traits or 7 varieties, 7 traits, characters or varieties of the pea plant. So the first one is height. So this pea plant is there are only 2 types of plant that is possible in a pea plant. Either the pea plant will be tall in height or either it will be dwarf. You will not see any pea plant which will be intermediate height, somewhere in between. No, either a tall, there a definite height is there which we consider as tall, and very short, very short dwarf. Then color, either the pea plant will have a purple color or it will have a white color. So because of this ease, differentiating color, or we say as contrasting characters, either it's like black or white. Okay, it's like true or false type, nothing in between. So this becomes easier, because if he chose any sample which was showing somewhere in between characters, it becomes very difficult to uh, observe and get conclusion and interpretation in genetic experiments. So the flower color either is purple or white. Now you can see this table talks about the dominant. So all these are the dominant characters and all these are the recessive characters. Okay. Okay. Flower position either axial or terminal. Okay. That is, this is the plant. So terminal is at the top and axial is somewhere in between. pod shape either inflated or constricted. Inflated is it is so like there is air inside the pocket of the pea pod, and constricted means like flat. Okay, there is no air, so constricted. You might have seen when peas are brought from market at your home, so you will see some of the pods are inflated. That is, they are swell and nice round in nature, and some are very flat. Okay. Then also you might have observed the pod color. This pod color. Okay. Some are greenish and some are yellowish. So green is dominant and yellow is recessive in nature. Then also seed shape, that is the peas which are there inside the pod. You might have seen green round color. So yes, yellow round colored, and then either round or then another opposite trait is wrinkled. Okay, shrink, and color of the seed yellow or green. Okay, either it can be yellow or either it can be green. So these are the 7 contrasting traits or 7 pairs of true breeding of the garden pea variety, which is Physarum sativum, that Mendel chose. Okay. So how did he carry out the hybridization experiment? Basically, he selected 2 plants with contrasting traits. Let's say a purple color flower plant and a white color flower plant. Now, out of the 2, for one plant he did emasculation. What is emasculation? Removing of the male part. This we had done in the first chapter, reproduction in plants. We discussed about emasculation in detail over there. If you have not checked out the video, please go and see that video for better understanding. So emasculation is removal of the anther, that is the male part, male reproductive part. So what will happen? The flower is only left with the female part. So you can dust the pollen grains from the anther from another flower of another plant onto this emasculated flower. Okay, so removal of anther of one plant to avoid self-pollination. So this was done to avoid self-pollination because you want different varieties. Okay, Mendel wanted this. Then collection of the pollen grains from other plants, that is the male parent, and then it is transferred to the female parent. So pollination is carried out. Obviously, this is what? Not natural pollination. This is man intervened. So this is artificial pollination. Okay, and then they are allowed to reproduce, and offspring will reproduce, and then the offspring are collected and studied. So collection and germination of seeds to produce offspring. So this is briefly about the Mendel experiment. Fine. Now we'll begin with the monohybrid cross. Okay, so let's begin with monohybrid cross. Mono stands for single trait. Okay, hybridization, now we know when we are crossing 2 different parents, two different plants with 2 contrasting traits, and we are trying to see what are the characters that will be inherited by the offspring. Okay, so monohybrid stands for a cross involving 2 plants differing in one character pair. Okay, that is what I have just mentioned. For example, if you will see over here, the flower color, one parent is purple, is having purple flower, one parent is having white flowers. Another example, you can say you can take a plant which is having yellow seeds and yellow color seed, and another plant you can take which is having green seeds. So similarly, you can take any of these combination. Okay, either tall or short plants like that. So a cross which will have 2 plants which will differ only in one character. So you can take either the color of the flowers, one purple or one white, or the height, that is tall or dwarf. You can't take one one plant is having purple flower and the another is having round seed. So they will fall under same category. You have to have dominant and recessive characters. Okay, so example, Mendel cross purple and white flowering pea plants to study the inheritance of one gene. So at a time, you will be able to study only the inheritance of one gene. Okay, so how do we begin? Let's say we take a parent plant, 2 parent, one male parent and one female parent. Let's say this is the female parent and let's say this is the male parent. Okay, So this is tall, this is dwarf. You can see the genotype. You can see two capital T and two small t, homozygous condition. So the gametes will first segregate. So what is the type of gametes? Or both of them are capital T. So you will just write once capital T, and both of them are small t. So you will write the gamete small t. Okay. So this is the first step, that is selection of the parent. A cross involving two plants differing in one character pair is what is monohybrid cross. So pure tall and pure dwarf. Okay, both of them will produce homozygous. Both of them are in homozygous condition. They will produce what? They will have two types of gametes which will be produced. So this one will produce a capital T and this one will produce the small t. Okay. Yes, these are the gametes. Now this will further be, crossed. Okay. So this is how the F1 generation will look like. So the parent had two capital T and two small t. So one small, we can write two times also, no problem. So I will write it separately because the gametes will be written separately. Then this can combine with this, giving rise to capital T, small t. This can again combine with the another plant, giving rise to capital T, small t. Then this with this, capital T, small t. Then this with this will give you crossing up. Okay, capital T, small t. So all the progeny, this is the cross between the, let's say, female parent and the male parent. So all the progeny that you see in first generation. So this was the parent and they have their first generation. Let's say they have four offspring. So all of them are what? You see the same genotype, right? They all are what? Heterozygous. Capital T, small t, capital T, small t, capital T, small t. This is the genotype. What is their phenotype? What will be they be? Tall or dwarf? So we just saw that capital T and small t, which will be expressed. The capital T will be expressed. That is, all the plants will be tall in phenotype, and the genotype will be capital T, small t, capital T, small t. Okay. So this is how first generation will look like. Okay, let's have a look in the picture. This. Now first generation, now first you got the first generation, now first generation will again undergo second generation, that is F2 generation. Okay. So in first generation, you all got heterozygous tall plants. All heterozygous tall plant. Now for F2 generation, then what will be the parents? So for F2 second generation, the parents will come from first generation only. No? All the parents, all the offspring in the first generation are this. So both the parents for F2 generation will be hybrid tall. Okay, they will have one capital T small t type of gene and they will be tall in the phenotype. So both the parents will be same for F2 generation. Okay, you will take the parents from the F1 generation only for carrying out F2 cross. Okay, so this is the F1 generation. You will write the gametes separately, one capital T, one small t, one capital T, one small t. And this is how what you will get in F2. Okay, so this is a summary. Let's do the F2 calculation over here. Okay, what will be the F2 calculation? Come on. So F1 you got all heterozygous tall. Now you are doing F2 generation. Basically, you are doing selfing. You are trying to crossing the progenies among themselves. Okay, you just choose one as the female and the another as the male, and you are crossing or selfing among themselves only. Whatever you got in the first filial generation. Filial stands for son. Okay, so it's a foreign name. Then gametes you will write separately. So for this, you do this checkerboard method. You draw a box and you write the gamete separately. So horizontally and vertically. Yes. So let's say this is the male represented and this is the female represented. Now you will do the multiplication. So first you will do this into this. Capital T into capital T will give you 2 capital T. Then this into this. So capital T into small t will give you one capital T, one small t. Now this into this again. So one capital T and one small t. Always you will write the capital letter first and then the small letter, and then one small t into another small t. So one small t into another small t. So this is what you will get in F2 generation. What you will get F2 generation. Now F2 generation is slightly different from what you got in F1 generation. In F1 you got all hybrid condition and phenotype was tall. Now here you will see in F2 hybrids are only two, this and this. So they are hybrid tall. Okay. What do you see? These are parental combination that you saw in the uh, F2 generation. So this is resembling the one of the parent and this is resembling another parent. So homozygous condition are seen in F2 generation back. Okay, like the parents one. Okay. So you got homozygous tall 1, then heterozygous tall 2. Yes, you got homozygous tall 1, heterozygous 1, 1, 2. Here it is represented. And homozygous dwarf. How many you got? 1. Total you get only 4 possible combination in the F2 generation because you're considering only one trait. That is the height of the plant. Okay, so this is how you will represent your genotypic ratio. Genotypic ratio is 1 is to 2 is to 1. 1 is to 2 is to 1. And phenotypic ratio, out of the 4 plants, these 3 are tall irrespective of whether they are homo or hetero. The phenotype will be tall of this and this and this. So 3 tall plants and 1 dwarf plant. So phenotypic ratio, you will write it as 3 is to 1. Clear? So this is how cross is represented. Monohybrid cross. Okay, I will just put, so you have a clear look at this. 2 capital T, 1 capital T, 1 small t, capital T, 1 small t. Okay. This is the complete picture of a monohybrid cross. You should be knowing the genotypic ratio and you should be knowing the phenotypic ratio. You have to memorize this. This can come for 4 to 5 marks for whole representation. Clear? So let's conclude this. So here Mendel saw that there is no in-between characters. Okay, all of them were either tall or either dwarf in that example. Here if you take the color, so either it will be purple or white. No, somewhere in between like pinkish or something will not be, was not observed. Then each trait is intact and discrete. Okay, so 2 alleles will be participating and individual trait gives individual characters to the progeny. Okay, for each pair, one trait was dominant and the other was recessive. So in the first generation, it was seen that all of the first generation plants showed dominant character, though the and the genotype was heterozygous in nature. This is what was observed by Mendel after doing monohybrid cross. Okay, pairs of the alternative traits examined was segregated among the progeny of a particular cross. Okay, that is the pairs that you got in the F1 generation, okay, were then used for selfing and were chosen as parents to carry out F2 generation. Okay, and this is how you got the phenotypic ratio. 3 were purple. For in this example, they are purple in color, and 1 is white in color. In the previous example, 3 plants showed tall phenotype and 1 dwarf phenotype. Okay, so the ratio of dominant to recessive was 3 to 4. That is 3 out of 4 plants showed dominant trait and only 1 showed recessive trait. So alternative traits were expressed in the F2 generation in the ratio of 3 to 4. That is out of 4, majority were showing dominant trait, that is here in purple color, and in the previous example tallness, and the only 1/4, that is 1 plant out of 4 showed the recessive character. So these are the outcomes or conclusion that were drawn from the Mendel's first monohybrid cross. Okay, so that's all for this video. In the next video, we are going to do the dihybrid cross, and then we will do the law of independent assortment, back cross and test cross. Fine. Before beginning this, we will also go through the laws of dominance and laws of laws of segregation, which will come from monohybrid cross only. So we are going to touch upon monohybrid cross with respect to the laws, and then we'll continue with dihybrid cross. So thank you all for this video. Take care. See you in the next video.